So welcome to another video tutorial on history of microbiology. So in which we'll be studying about another two great scientists, that is Joseph Lister and Robert Koch. So let's get started. The first scientist is Joseph Lister. His period was 1827 to 1912. Joseph Lister was an English surgeon and developed antiseptic surgery and pure culture techniques. So yes, he was a person who's very well known for antiseptic surgery. Okay, so antiseptic surgery is a process of destroying germs that caused infection before and after surgery. Okay, so I think maintaining antiseptics is one of the most important aspect related to surgery, right? So even if the surgery is performed in a very good manner, and if we don't prevent the germs which is causing infection, it's of no use. It may even take the life of a person. So right, so maintaining antisepsis is very much important. Okay, so this was not known to people before, so it was Joseph Lister who introduced antiseptic surgery. Okay, so going back to the history, I think less than 200 years ago, hospitals looked very different than they do today. Okay, so and hospitals were called as home or houses of death, and infections were more found in hospitals itself. Okay, and their doctors or patients didn't know what is the meaning of sterilization, and doctors used to not even wash their hands before dealing with their patients. Okay, and all the surgical instruments, no, they used to not sterilize, they used to not even wash, and it was completely filled with blood, dried blood and sawdust and all those things. And hospitals also had one of the bad smell. Okay, so and this was how the hospitals was, and people used to not clean hospitals, and the bandages were thrown there and here. So this was how it looked. Okay, so but he was a person who completely changed the look of the hospital, as well as he is the person who completely changed the method of surgery. Okay, so which made uh, hundreds and hundreds of surgeries successful. Fine. So Joseph Lister was born on April 5th 1827. So yes, he was born on April 5th 1827, and in 1861 Lister became surgeon at the Glasgow Royal Infirmary. So yes, he started his career at Glasgow Royal Infirmary. He performed and reported that 45 to 50% of his amputation cases died from infection in his male accident ward between 1861 to 1865. So yes, he was a surgeon in the male ward. Okay, so and he used to he what he noticed was 40 to 50% of his amputation cases died. Okay, now what's his amputation? Amputation is a surgical removal of a body part, that is especially legs or hands. It's a surgical procedure of removing legs or hands because of some injuries or infection. Okay, because of some accidents or gangrene, and there is an infection in that part of the body, it shouldn't spread to any other part, right? So it was a surgical method of removing that part. Okay, so that is amputation. So what he reported was 45 to 50% of his amputation cases died from infection. So infection was the biggest killer that time. Okay, and people didn't know where was the infection coming from. Okay, what caused infection? They didn't have an idea during that time. Okay, so Lister read the work of Louis Pasteur on the cause of fermentation of beer and milk bean due to living organisms. So that's when Lister would read the book of Louis Pasteur. Okay, and he would know that fermentation of beer and milk bean due to living organisms, that is there are some living organisms that is causing fermentation of beer and milk. He got to know the work of Louis Pasteur. Okay, then So yes, after getting to know about Louis Pasteur's work, so Lister also will think that yes, there might be germs, okay, in and around the surgical area, and that may be the reason which is causing infection. Okay, so what he started doing was he used dressings soaked in carbolic acid to cover wounds, and the rate of infection was vastly reduced. So yes, he started using carbolic acid in his surgeries. So what is this carbolic acid? So it's a phenol, okay, which is famous as antiseptic or disinfectant. Okay, that means it used to remove or kill the microorganisms around. So what he used to do was he used to completely soak the dressings in carbolic acid, and then he used to dress them after surgery. Okay, so and this actually made the rate of infection and the rate of the deaths to be reduced. Okay, and then he began to use carbolic acid to dress or to protect the area of operation from infection by the surgeon's hand and instruments. So yes, later he started using them on surgeon's hands also, and all the surgical instruments also. Okay, so in 1865 Lister operated on a 11-year-old boy who had suffered a compound fracture. The wound was cleaned with carbolic acid and covered with a dressing soaked in acid. So yes, so in 1865, so first he'll operate a 11-year-old boy who had suffered a compound fracture. Okay, then he will completely dip the dressing in carbolic acid, and then he will dress it on the site of the surgery. Okay, and at regular intervals, the wound was cleaned and dressed, and within 6 weeks, the wound was completely healed. So, yes, at regular intervals, so it was again and again cleaned, okay, and it was dressed again. So, and he noticed that within 6 weeks, the wound was completely healed and the boy was completely alright. Okay, so and then Lister successfully introduced carbolic acid, now known as phenol, in 1865 to sterilize surgical instruments and to clean wounds, which led to a reduction in the post-operative infections and made surgery safer for patients. So yes, so after his discovery, so he introduced that carbolic acid can be used to clean wounds. Okay, and this is the one which made the infection to drop down, and it is safer for patients. So he is the one who introduced that, and he is the one who told that there is germs in and around, and germs are the one which would cause infection at the site of injury, at the site of surgery and all, and maintaining sterilization and disinfecting these areas are very 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 important. Okay, then yeah, between 1864 and 1866, before the use of antiseptic treatment, 46% of the Lister's surgical patients died. In contrast, from 1867 to 1870, only 15% died. So just see the contrast here. Okay, so that is before using antiseptic method, no, 46% of his patients died. But after following antiseptic precautions, no, only 15% died. Okay, that was actually it's a vast change, right? And hundreds and hundreds of patients are saved here. Fine. So and this was one of the major contribution which is given by Lister. And later he also developed a method of repairing knee caps with metal wires. As he was a surgeon and he was operating, he knew more about all such things. He also will develop knee caps with metal wire. Okay, and then Listerine mouthwash is named after him for his work in antiseptics. Yes, I think all of you are known about Listerine mouthwash. So this is actually named for his work. Okay, so this is about Joseph Lister. So next scientist is Robert Koch. So his period was from 1843 to 1910. Yeah. So Robert Koch was born on December 11th, 1843. So yes, again he was one of the greatest scientists who's discovered and who's given a lot of contributions to the field of microbiology. So we'll study some of the important contributions here. And he was born on December 11th, 1843. So he was a German bacteriologist. Okay. He isolated causative agents that are responsible for causing disease such as anthrax, tuberculosis and cholera. So yes, so these were one of the deadliest diseases during those time. So he is the one who isolated the organism which caused these diseases. Okay, so that is for anthrax, it is Bacillus anthracis. So he isolated this causative agent in 1876, and for tuberculosis it is Mycobacterium tuberculosis. So he isolated this organism in 1882, and for cholera it's Vibrio cholera. So he isolated this in 1883. Koch found that the disease could be spread by the blood of infected animals. So yes, he also told that the disease could be spread by the infected blood. Okay. Then further, Robert Koch invented streak plate technique. So now what's this streak plate actually? It's a method to isolate individual colonies from the mixed cultures. Okay, it's generally done by diluting inoculum across the agar plate's surface. So as you can see in the picture, so on top of the agar medium, so it is streaked. Okay, the inoculum is streaked, and this method, streak plate method, was given by Robert Koch. And he also introduced gelatin as a solidifying agent in nutrient media. So yes, he is the one who used gelatin. He told that gelatin can be used as a solidifying medium, on top of which so you can culture the plates. Okay, then. Okay, so in this slide we'll study about Koch's postulates. That is, Koch actually will give a set of criteria used to establish a causal relationship between a microbe and a disease. That is, whether this particular microbe is responsible for the disease or what. Okay. Now the microorganism that is responsible for the disease should be constantly present or found in the lesions of the disease. So that means, now say imagine there is an animal who's got the particular disease. Okay, so when you isolate microorganisms from that animal, no, so that particular microorganism has to be constantly present every time. Okay, then pure culture of the organisms can be isolated from the lesions of the disease. So yes, and then you should be able to make pure culture out of that organism, and then inoculation of the pure culture in suitable laboratory animal or experimental animals should reproduce the same disease. So that is, when you inject the microorganisms from this pure culture to another animal which doesn't have a disease, that is to a healthy animal, so that particular animal also has to get the same disease. Okay, only then you can confirm that this microbe is responsible for the disease. So you can see in the picture very clearly. So microorganisms are isolated from a dead animal. So there's a dead animal, so it's dead because of some disease, and microorganisms are isolated from that particular animal. Okay, and here in this picture you can see the microorganisms are grown in pure culture. The organisms here are grown into pure culture. Okay, that is to a single type of microorganism. It's pure culture, and then you will identify that microorganism. Okay, you will observe them through microscope and you will identify which is the organism, and then this microorganism will be injected to the healthy animal. Okay, see, the microorganisms are injected into a healthy animal. So when you inject this microorganism to the healthy animal, the disease is reproduced in the same animal. Microorganisms are isolated from this animal, so same disease has to develop in the healthy animal also with the symptoms. Okay, and then again from this animal, when you isolate the microorganism, when you grow into pure culture, and then when you identify them, no, it should be the same organism. Okay, so which you found here, the same organism has to be seen here also. Only then you can confirm that this is an organism which is causing the disease and it's responsible for the disease. Okay, so this was given by Koch.